Great. So it is now 9:01. We'll begin our Benchling Bootcamp session right now. So hello everyone. Good morning. Welcome to the first Benchling Bootcamp of November. Today we'll be going over the basics of the Benchling Notebook. A quick agenda of today's webinar. We'll start out with welcomes and introducing the members of the Benchling team. Then we'll go through some slides that overview the Benchling platform, as well as cover projects and permissions as they relate to the notebook. We'll be spending the majority of our time in platform demoing the Benchling Notebook, and we'll wrap up with some announcements and instructions for you to access our recent product updates, as well as resources to help you familiarize yourselves with the wide range of functionality that Benchling provides. So first of all, just to introduce everyone on the Benchling side, my name is Freeman. I'm a CSS who will be leading today's training exercise. We also have Alex, Chin, Stefan, and Meredith as panelists on the call, and we're all members of the Customer Experience Organization here at Benchling. And all of us are here to provide answers to your questions and support you all in your use of Benchling. Today, we'll be leveraging the Q&A button to address any questions you might have during the session. So feel free to ask any questions you have during this exercise via the Q&A button shown here. Another feature we'll be using are Zoom polls, which will come up as new windows on your screen. And these are generally open-ended questions that will help us gauge how different users are utilizing the Benchling Lab Notebook and provide specific examples relevant to your particular use cases. We'd love to hear about how you or your team could use Benchling. This information could also help you learn about other various ways to use Benchling with your peers. So we'll be giving a minute or so for you all to send a response to the Zoom polls, which will be in the form of a multiple choice question that will pop up on Zoom. Let's move forward. Just to introduce the Benchling platform, it's composed of these 7 major applications. And while today's training is focused on the notebook, you can see here, we do offer custom trainings for each of these applications. So if from this webinar, your team is interested in a training around, for example, the molecular biology or the registry tools, we'll provide some ways at the end of this session to get you connected to our training team at Benchling and arrange those for you. So back to the notebook, we think of it as a central source of truth on the Benchling platform, meaning that it can encapsulate all parts of your research workflow, from observational notes to experimental protocols. So as we get into the more advanced features in our second session following this, you'll find that it can sync with the other applications in Benchling with structured tables such as registration or transfer tables. And these applications are all interconnected and integrated with one another because of our united data model, which is able to model biological entities such as DNA or protein sequences, as well as notebook entries and results data. And these different forms of data are represented similarly across different applications, which creates a united platform that aggregates all of this information. So now we're going to dive into how data can be stored and managed efficiently in Benchling, particularly in the context of the notebook application. So we'll start talking about projects, folders, and notebook entries. And within every application on Benchling, we like to think of the primary form of data that lives within an application. So for the notebook application, these would be entries. I can begin by explaining the hierarchy of data organization from the bottom here with entries. These entries are text editing documents that are used to plan experiments, execute research, and they're home for both your observational notes and your experimental protocols. Moving up from notebook entries, we have folders, which are very similar to your file explorer on your computer. So it's simply a way for you to organize your notebook entries or other types of data within the notebook. In contrast to projects, permissions are not specified at the folder level, so folder-specific access is not available. And that brings us finally to projects, which is the highest level of organization in the Benchling Notebook. They can be organized, for example, by research group, by individuals, or by target of interest. Permissions are set at the project level, so any folders and entries within a particular project will take on the permissions of that project. As a best practice, we do recommend that you minimize the number of nested folders to reduce the number of clicks and efficiently allow relevant data to be found. To support a minimalistic data structure, we'll be showcasing Benchling's robust search capabilities with a tool later called Global Search, which allows you to find your data quickly and consistently. And finally, let's take a look into what types of permissions that these projects can take on. Permissions come in 4 different types which stack on one another. So beginning with the lowest level of permissions, we have the none permission that you see here, which means that users cannot read or create data within the project. Higher than none is read, in which users can view but not create data. Users with write permission are able to view and create data, such as notebook entries within the project. And the final level of permissions is admin. So a user has all the permission levels of the previous settings. They're also able to add collaborators to a project and adjust their permission settings for that project. One item I want to point out is that permissions for a project can also be set not just for individuals, but assigned to teams or organizations for easier grouping. So for example, if I have a microbial engineering team with me, Stefan, and Chin, you can assign write access to our team for a particular project. And me, Stefan, Chin, and whoever else is in the ME team will also have write access to that project. Another concept that rounds out this topic is per entry permissions, or authorship. Essentially, when a user creates a notebook entry, they are by default the author of that entry, and thus they're the only user that can edit their created entry. I'll be showing later on how to add a collaborator as a co-author in a notebook entry, which will allow them to make changes to a colleague's notebook entry. And just as an example for why all of this is important, we see many use cases where we have external collaborators, such as CROs, temporarily needing access to very particular sets of data. So for these cases, you can generate a project just for them, in which the data that goes into that project is carefully selected and reviewed. So of course, this type of data management and organization is also used internally by giving write access to particular teams and read access to teams that might not or should not contribute data to particular projects. Great, so now that we've gone over the concepts behind the Benchling Notebook, I'm going to leave the slides for a bit and dive in platform for our demo to see how we can leverage the notebook to support all aspects of data capture. So as a cloud-based platform, I don't need to download any desktop applications, and I can immediately navigate to Benchling by opening a new tab in my web browser. Before we get into our example experiment, let's orient everyone to what we're seeing within the Benchling platform. So what we see here is a Benchling notebook entry titled ELISA for bacterial toxin detection, which will be the exercise template that we're going to be covering today. Majority of the space that you see here is what we call the workspace, which is an area on the right-hand side of your screen that will be populated by any type of data that you open in Benchling in the form of tabs that you can see here, similar to a web browser. One item I want to point out here is at any time during your use of Benchling, if you run into any questions or you're unsure about how to perform a certain action, you can click into this blue question mark here, where you'll be able to see options to either chat with a customer or a customer success representative such as myself on the Benchling team, as well as the option to search through any of the comprehensive help articles via this graduation cap icon here. Additionally, if you want to access some settings, you can click on the avatar in your lower left corner of your screen, where you'll see settings here to configure your name, your email associated with your account, as well as your picture in Benchling. So again, for all settings related actions, remember to click on your avatar here on the bottom left of the screen. Great. So now to walk through how we navigate through the Benchling platform, I'd like to point out this navigation bar here on the left, which constitutes some major applications in Benchling. So I'll walk through a few of these key icons. First, we have this jellyfish logo, or Jeffy P, our Benchling mascot. This will be your home button. which when I click on it, will open up a window that accesses any recently opened entries you can see here or work lists. I can press on this caret here to expand, which will open up my calendar, which populates with any notebook entries that I'm currently working on. As you can see, these notebook entries can be associated with particular dates. This kind of gives me a bird's eye view of what notebook entries I'm working on currently. To get a more granular sense of what you're working on within a particular entry, you can click into that entry, such as this one, and see checklist items that are pulled directly from the notebook entry. One perk of this feature is that this connection is dynamic. So if you check something off here, the checkbox within the entry will also be unchecked or marked as done, depending on what you do here. This is very useful for a quick step that you might want to step into the laboratory for, such as a nanodrop measurement, and you would like to mark it as complete quickly without having to find your entry, navigate to a particular checkbox within your entry, and mark it there. So I'll X out of this here. But overall, this highlights the usefulness of the calendar, both as an organizational tool to manage what you or other Benchling users are doing in the lab from a high level, as well as a way to track small steps and experiments from a granular level. So to minimize my calendar, I can press on this reverse caret here, and I can press on this home icon again to restore my full workspace. Great. So now I'll click on the projects icon, which will pull up the projects that are available to me. And that is shown with this briefcase icon right here. In this case, I have access to two projects, the antibody production project, as well as the bacterial toxin detection project. If you would like a particular project to always be at the top of your list, you can start a project by clicking on the star icon, which I've done so already. And of course, this would be much more applicable in situations where you might have more than two projects to sift through. But if I click into my bacterial toxin detection project, I can see what's inside of it so far. In this case, I don't have any nested folders, but I do have two notebook entries. And before we get into this notebook entry, I would like to call out that we use a type of navigation called breadcrumbs navigation in Benchling. So say, for example, from here, I wanted to return to the list of all my projects. Instead of pressing the back button on my browser, I can use this clickable projects button here to return to where I came from. For each nested folder that you're in, you'll see more of these clickable links, which will take you exactly where you want to go. For now, let's move forward and click into our notebook entry here, titled Anthrax Sample Collection. As you can see, it'll open up like a tab in a browser, and you'll be able to see the contents of the entry by scrolling. So of course, similar to Google Chrome or any other browser that you're using, you're able to click and drag these tabs like so. So to create new data, I'll press on this blue plus icon to open up a drop-down menu of items that I can create. So as I mentioned earlier, you can create folders as well as DNA sequence or other biological entities. But to create a new entry, I'll follow the dropdown to blank entry and click through. A new tab will open with the blank entry that I've created, where you have essentially a new canvas to record your observations or keep track of steps in a protocol. For today's exercises, I'm going to be working from a notebook template that I've created already, which is this entry that you're seeing here. We'll be going over notebook templates more in our advanced notebook training, but you can see that it's an efficient way to standardize your notebook entries, whether it's pre-populating an entry with a protocol that is commonly used in your lab, or just providing a backbone to guide users to capture data in a consistent manner throughout a research process, so no steps or observations are missed along the way. So I want to create more space for myself, so similar to how I minimize my calendar, I'll click on the projects icon again to increase my workspace, like so. So today, the entry that we'll be working from today is titled ELISA for bacterial toxin detection. And this alludes to our scientific focus for today, which is centered on infectious disease research. with a specific emphasis on anthrax, as well as the bacteria that causes it. We felt this was an engaging and interesting topic to model our webinar after, but I just wanted to point out that all the items covered here today are broadly applicable to other research settings as well. So some background on anthrax here. It's an infectious disease caused by a gram-positive bacteria, and cases have been documented on all continents, with the possibility of infecting any species of mammals. The CDC has categorized it as a Category A bioterrorism agent, which makes anthrax a significant topic of interest for infectious disease researchers across the country. A simplified overview of how toxin levels can be detected is shown by this figure below. The researcher begins with infected mice, which are inoculated with the bacteria that causes anthrax. From these mice, blood or tissue samples are isolated. These samples are then used to perform an indirect ELISA. Because studies have shown a correlation between disease severity and concentration of the toxin, this ELISA will be measuring the concentration of the anthrax toxin in these samples as our data points of interest. For this experiment, we're going to be utilizing a primary antibody that you can see here, specific for the antigen, and a secondary antibody that is linked to HRP. So this brings us to poll question 1, which will open up a new window on your screen. This helps us to gauge what you're looking for in terms of functionality. So for this, for the Benchling Notebook, we'll take a few seconds for you all to respond, about a minute or so. So please feel free to answer in this time. Again, this will pop up as a new window on Zoom. Great. Thank you all for your inputs. I'm glad to see a wide variety of functionality that you're all looking for out of the Benchling Notebook, both as an observation-based note-taking tool, but also as a source of truth for any experimental protocols. So hopefully, as we finish this training today, you'll be able to see both the flexibility of a notebook entry and its ability to be a singular source, both for qualitative observations as well as quantitative recorded data. So continuing with today's exercise, we'll see a set of preparation materials that we need for the ELISA experiment in the form of checkboxes that you can see here. As we're gathering materials, we can check off these reagents, which will also be checked off in my calendar, like I mentioned earlier. You're also able to see here that there's formatting options available for text in a notebook entry that is accessible in this formatting toolbar at the top of your entry. These options are similar to what you might see in any word processor, so you can bold, italicize, underline, change the text color, as well as the highlight color. You can also insert symbols such as Greek letters or degrees, and I'll demonstrate that by inserting a symbol. I'll do that by navigating to Insert, Symbol, and here I'll have a wide variety of different symbols to input. Great. So as we continue to check off these reagents, we may then want to specify which specific blood or tissue samples from our anthrax-treated mice I want to use for today's experiment. To define these samples, I'll access an application that I alluded to earlier called Global Search, and that is this magnifying glass icon found on the left navigation bar. If I click into this search icon here, I'm given different parameters I can adjust to narrow down my search results. I can specify the type of data I'm looking for, such as DNA sequence, amino acid sequence, etc. In this case, I'm looking for samples from my mice. If I apply this type filter, I'll see all the samples that I have in Benchling. I can further refine my search by applying filters. I'll specify that I want blood samples. So to do that, I'll go to blood, select sample type, and I'll look for blood. I'll apply this filter. And so now the results that I see are even more curated. I'm not interested in all of my samples, but specifically blood samples, which are the 4 that we see here. Of course, this is a simplified scenario, but Global Search is a powerful tool that allows you to locate and organize your work. And we'll go over this tool more in depth at our advanced training following this one. If you click into the expanded view via this caret, you can leverage bulk actions such as archiving or moving for even more flexible data organization options. So say I've decided to use one of these samples in my experiment. Let's say I want to use sample 001. I know it's a blood sample, and that's the one that I'll be using for my ELISA today. I can @mention them in my entry by typing @ and the first few words of the entry that I'd like to reference. So for example, say I want to find this sample 001. I'll type in sample 001. And so what this does is this creates a bi-directional clickable link called an @mention in Benchling. So if I click into it, it'll direct me into this samples page in the registry. As you can see, it's a blood sample. And if I scroll down to relevant items, I can see that this sample was referenced in my entry. To demonstrate how this link is bidirectional, I can click on this link, which will direct me back to the entry where I @mentioned this sample. In addition to being able to @mention registered entities, I can also @mention other notebook entries. Again, I'll type in @. Instead of searching for an entity this time, I want to search for my entry titled Anthrax Sample Collection. As you can see here, now this populates with this entry, which I also have the option to click through, which will again generate this bi-directional link. If I click into this entry, again, I can see if I go into this metadata, I can see that this entry was referenced just now within this entry. Great. So let's move forward with our ELISA experiment. Again, I'll click on this search icon to minimize my search so I can get more space here. To move forward, let's say we gathered our relevant materials, which is reflected by the checkboxes on our calendar and our entry. What we've also done so far is we've @mentioned and we specify the samples that we're going to use, in this case, sample 001. And we've also documented my collection of blood samples for the ELISA with this entry titled Anthrax Sample Collection. So say now we need to do some quick math to calculate our master mix values for our ELISA. We can do so with what we call an unstructured table in Benchling. What these are Excel-like tables that are useful for performing basic calculations with the ability to add a variety of functions that you commonly see in Excel from this function button here. So for example, if I want to calculate the total volume for a master mix, I could utilize the sum function or use the average function to find average fluorescence for a set of samples. We do have some formulas already populated within these cells. So when I change the number of replicates in this cell, for example, to 2, and press enter, the total volumes, as you see here, will automatically populate with our new volumes. That calculation is carried out simply by using the number of replicates that I've inputted here as a multiplier against the values in this volume per replicate column. Just a reminder that this notebook entry was created from a template. So these tables that you see here with pre-built formulas can be useful for calculations that you find yourself doing regularly, such as a PCR master mix table or an ELISA master mix table like this one. To insert another unstructured table, just click anywhere in the entry that you like your table to be. Go to Insert, and then Table, where you'll be able to specify the dimensions of your inserted table. So if I create a small 2x2 table here, I can go ahead and click through, and I'll have this 2x2 table. One thing I want to point out within this new unstructured table is I want to say that Benchling supports cross-table functions. So if I type equal here, I can set this cell equal to a cell, for example, this one. Go ahead and press enter in another table, like so. Another type of unstructured table in Benchling is a plate layout. So from my lab days, I would do the NADH glow assay with 2 96 well plates, with each well containing a different bacterial culture. I distinctly remember not only having to write down each sample that I use in my lab notebook, but also having to physically draw a sketch of the plate and map the components of each cell after the assay is finished. But with the benchling plate, I can solve both of these time-consuming and error-prone actions at once. So on this ELISA plate, I can see where I place my positive controls, as well as where I place my negative controls. Of course, the positive controls are the samples which are known to have the anthrax toxin, and the negative controls are samples which do not have the toxin. We can also then specify the particular samples that are going into the wells. To do that, I can click into a cell, and similar to how I had mentioned the sample earlier in my notebook, I'll type @ and then search for my sample of interest, in this case, sample 001. Again, I'll go ahead and click through this. As you can see here, now this bidirectional link is now present within this cell. So we know that sample 001 is in cell B4. Of course, I can click and drag to populate the other cells with sample 001. Great. So just to show everyone how I was able to create these plate layouts, I'll click into a place on my entry where I'd like the plate to be inserted. And similar to how we created our table, I'll first navigate to Insert, go to the well plate dropdown, and I can have the option to create a well plate with dimensions I would like. Wonderful. So that brings us to our second poll question, which asks, would it be beneficial for you to be able to map out a plate or a table without needing to open Excel every time? So I will go ahead and open this poll question. And again, please take a minute just to put in your responses here. Great. Thank you, everyone, for putting in your responses. So now we can continue with our ELISA experiment. So we've gathered our reagents. We specified which samples we're using, as well as where on the plate the samples will be going. We'll continue with our experimental protocol here. As I finish my incubation, I'll check off these items here with my primary antibody, secondary antibody, and now say I'm on the last step of my experimental protocol, in which case I'm measuring my absorbance at 450 nanometers with my microplate reader. So now I'm collecting data that I'm interested in for this experiment. So as I mentioned earlier, Benchling entries are great both as an experimental protocol, as we see here, but also as a place for capturing recorded data. In this case, I'll insert an Excel spreadsheet of my data, and I'll do that by clicking on Insert, just as we did with the table and plates. And instead, I'll select the attachment option here. So here, I'll have a space to upload a file from my computer, and I'll attach an Excel file of my result into this entry. I'll go ahead and click Upload a file here, which will bring up a folder I have called Training Materials, and within it, an Excel spreadsheet called Example ELISA Data. So one item that I want to point out here is that this Excel file that is uploaded onto Benchling is a duplicate of the file that I have saved on my computer, meaning that now it's a backed up file on the Benchling cloud, so I don't have to worry about where it is on my computer. I can see a preview of the Excel sheet from within my notebook entry by pressing on this icon right here. So as you can see, it'll pull up my example ELISA data that you can see here. If I go back to my notes, I can also have the ability to download this attachment back into my laptop by clicking on these 3 lines and pressing download here. So if I download this attachment back into my laptop, I would have the ability to open it in its native application, which in this case would be Excel. One item I want to point out here is for our advanced notebook session, we'll be covering a tool called Benchling Sync, which will streamline this process of editing file attachments. So be on the lookout for that as well. So now I'm going to open up this preview again. Let's say I'm looking through this data and I see an interesting outlier here I would like to discuss the results further with a colleague. What I could do is mention them in this entry, similar to how we mentioned a notebook entry already, and similar to how we mentioned a biological entity. So what I can do is, again, I'll press @ and I'll search for my colleague Chin, who is also on this call. I can find Chin here, and again, I can click through, and this will tag Chin on this entry. This will send Chin an email notification with a link to the entry that I mentioned them in. So of course, be mindful in your mentioning, so your teammate isn't getting 20 emails a day from mentions. But I want to point out that this tool is very powerful in communicating with team members that you want their eyes on a particular entry. I remember that anytime a colleague and I want to discuss the results from, for example, our NADH glow assay, it will start out with an email that I have to attach the file to and multiple back and forths on one thread. And then we would have to forward the whole thread to our PI, which became quite a hassle. So this simplifies that communication to a simple @mention. Perhaps I can even say @Chin, take a look at cell B4 and let me know what you think. For example, if I had a particular data point that I was interested in. One item that I like to point out here is that @mentioning will not override the permissions of the project. So if Chin doesn't have at least read access to the project this entry is in, she'll receive an email and a link to that entry, but she'll be unable to see the entry because she doesn't have the permissions for that project. In the case that I also want Chin to have the ability to edit my entry, I'll have to add her as a co-author on this entry, which brings us to our third and final poll question. How often do you collaborate together with a colleague? So again, I'll launch this poll question, our last poll question, and please take some time to respond. Great, thank you everyone for your responses. It looks like some users collaborate, some people don't, but we can kind of take the time here to mention how easy it is to collaborate with a teammate on Benchling. So I've mentioned Chin, but now I like to add her as a collaborator in my entry. To do that, I'll switch to the metadata tab from the notes tab. So currently I'm in notes, I can switch to the metadata tab here. And here I'll be able to view and edit many properties of my notebook entry, such as its location in Benchling, as well as its authors. I'm also able to see when it was created and this unique experimental ID that is assigned to my notebook entry. So to add Chin as a co-author to my entry, I'll click on this pencil icon next to authors here. And I'll search for an additional user, such as Chin. I'll go ahead and click on this dropdown. I'll press this blue check mark. And now Chin is able to edit this notebook entry. So I'll head back to my notes here. So now that Chin is a co-author on my notebook entry, it might be useful for both of us to be able to visualize where and when each of us are making changes to this entry. To do that, I can toggle the setting for that by navigating to this gear icon and selecting this timestamps option here. When I select this checkbox, I'm able to see all the different changes that are made to this entry and who is making these changes to this entry. I also have access to other features such as spell check and print layout, as well as the table of contents feature. When I enable table of contents, this box now appears, which populates with headers in my entry. So for example, at the very bottom here, I create an example header. I'll go ahead and highlight all of this and make this a big header. I can see that reflected in my table of contents feature. So if I click into example header, it'll bring me to exactly where I want to go in my entry. It's very useful if you have a long entry and you want to have a way to navigate quickly to exactly where you want to go. You can create these headers, and with the table of contents feature, you'll be able to navigate there. One thing I do want to point out here is that tables and well placed are also reflected in this table of contents feature. In the same vein as the timestamps feature, you can also find versions of your entry at different times by clicking on this clock icon on the right sidebar. From here, you'll see multiple iterations of this entry at different times, and note which user was responsible for the change. So for example, if you click into one of these iterations, such as this one, you'll be able to see what that version looks like and have the option to reset to that particular iteration. You can also clone from an older version. For example, if you reach a fork in an experimental workflow, which essentially duplicates a version into a new notebook entry. So I'll reset back to this current entry here. And all of this functionality that I just showed here is to encourage users to be creative and active in their data capture, with the assurance that any accidentally deleted data or mistakes can easily be undone with a Control-Z, or even a reset to a 5-minute-old version of the entry. So some additional features that I like to show everyone as we round out this training session is how to perform actions on entries in a project, as well as set project permissions if you are an admin of a project. So if I click into my briefcase icon and bring up my projects list, I can right click on the bacterial toxin detection project. When I do that, I'll be able to see a set of options here. I'll go ahead and click into settings. When I do that, I'll have the option to edit my name, give my project a description, as well as change different groups or users that have access to this project, as well as permissions that are given to them. So in my case, I'm Benchling support, as you can see here, and I am an admin for this project. I can edit the permissions for the members of the LISA team, for example, from write to read. I can also add my colleague Chin as an individual user to this project. Great. So to perform actions on an individual entry, however, let's click into an entry. So within my bacterial toxin detection project, I'll click in, and we have 3 entries in here. So say I want to do an action on a particular entry. What I can do is similar to how I got into project settings. I can right click on one of these entries, for example, this untitled entry here. I'll see a set of options also available, such as moving it to a different location, copying it somewhere, renaming it, or archiving it. I do want to point out what archiving means on the Benchling platform. In short, we archive instead of delete data on Benchling to ensure that data is not accidentally or erroneously deleted. So that means even if I archive this entry or any notebook entries that you archive, they're still searchable and recoverable whenever you might need it again. And again, I'll minimize this by clicking on my projects icon here. So that brings us to the end of our in-platform exercise. And just to recap here, we took on the role of a scientist interested in the research of anthrax, and we designed and executed an ELISA experiment to determine the concentrations of the toxin in various animal samples, in this case, mice samples, blood, and tissue ones. So we also saw the ability to utilize an entry as an experimental protocol with dynamic check marks that link to your calendar, as well as demonstrated the feature of mentions seen here of biological entities, notebook entries, and even other Benchling users via @mention. We also showed that you can use unstructured tables in your entries, both to pre-format tables, for example, for different experiments, also to map out a well plate, for example, with this exact ELISA experiment. We also went over how to attach external files in an entry, as seen here, as well as the ability to add a teammate as a co-author. And we do that via the metadata tab here under the authors tab, as well as revert our notebook entry to previous versions. And again, just to showcase that real quick, you can do that by pressing on this clock icon, in which case you'll have access to all the different iterations of this notebook entry. Great, so now I'll jump out of the platform and back into our slides. And we do want to call out that our platform is constantly evolving and adding new features, some of which are the newest ones, which are showcased here. We also release monthly notes, I'll click into that here, that show product improvements and features typically suggested by users like you. So these notes, as you can see here, are also emailed out once a month. So if you want to keep up to date with how we are improving our product, do be on the lookout for these emails. Going back to our slides, we're really excited for you all to be here. And we're really looking to continuously improve our training methods and your experience. So right now, we're going to send out a quick survey, which will be sent out via the chat. It's really short, should only take about a minute to complete. And we'd love to hear your feedback on this. So please just take a short time to respond to this survey. It will be appearing in the chat shortly. So please click in, answer the questions, which will help us design sessions that are more useful for you in the future. Great, so I'll jump forward now to letting you know about our training resources that are available. We do have a comprehensive list of help articles that are available on help.benchling.com. I'll go ahead and click into this here. As you can see, we have a variety of help articles here. They're all very comprehensive, very helpful. They're separated by topics. You can search for anything that you would like here, and you're likely to find an article that helps address what question you're looking for on the Benchling platform. Again, we also have our in-app chat, which is available via this blue question mark here when you're in platform. And lastly, one thing I want to point out is that our Anthrax training today was an example of a custom training in a particular research vertical. We also offer tailored trainings for all major Benchling applications, such as registry, inventory, or request applications across an even greater variety of research verticals. So if anything like that is of interest to you in a field that you're working in, please feel free to reach out to our training team at training@benchling.com. And they'll be able to connect you with any custom training needs that you might have. So with that, thank you everyone for joining us for the first session of our November Benchling Bootcamp. We love to see you at our next session. I alluded to it quite often today during this exercise. So that will be covering advanced techniques that make you even more of a Benchling Notebook expert. And we love to see you there. Thank you, everyone.